I'm Dr. Sehrish Zaffar and I welcome you to Key to Pharmacology. Within journal Pharmacology, Pharmacodynamics, बच्चों को मुश्किल लगता है. What is pharmacodynamics? First of all, we need to know the definition. Pharmacodynamics is all about the physiological and the biochemical changes that are occurring inside our body due to a specific drug that has been administered. एक drug, whenever it gets into our body, उसके बाद उसने क्या action produce किया, किस तरह किया, उसका body के ऊपर क्या effect आया, useful effect आया या कोई toxicity आई. That is all covered under pharmacodynamics. When we talk about the movement of drug in the body, कि drug administer कहाँ से हुई, drug absorb कहाँ से हुई, कितनी amount में हुई, उसकी bioavailability कितनी थी, उसके बाद वो कहाँ distribute हुई, that is all the kinetics. जब ये सारे processes, when they are in their places, the drug finally reaches its site of action. वहां से आगे start हो जाती है pharmacodynamics. Now, once the drug has reached where it wanted to be, या where it was supposed to be, then how it is going to affect that system? What is the receptor that is going to act upon? What are the cellular mechanisms that it is going to activate or inactivate? What are the pharmacological effects that it is going to produce? If those pharmacological effects are useful, they are called clinical uses of that drug. If those pharmacological effects are harmful, they are called adverse effects or toxicities of that drug. All of this is included within the domain of pharmacodynamics. Before we hop on to the more difficult topics of receptors and agonists and antagonists, it is important कि हम एक basic revision कर लें cellular mechanisms की which are involved in signal transduction. These signaling mechanisms involve 5 different types of receptors, broad categories. They can be intracellular receptors, they can be transmembrane enzymes, they can be cytokine receptors, they can be ligand-gated ion channels or G-protein coupled receptors. Intracellular receptors, as their name indicate, the receptors are present inside the cell. Now, intracellular receptor का मतलब ये है कि receptor cell के अंदर पड़ा हुआ है, which means that the ligand, the drug which is going to act on that receptor, उसको पहले cell membrane cross करनी है, सेल के अंदर आना है, फिर अपने रिसेप्टर को ऑक्यूपाई करना है. फॉर दैट टू हैपन देयर आर सर्टेन प्रॉपर्टीज जो उस ड्रग के पास होनी चाहिए. द प्राइम प्रॉपर्टी इज लिपिड सॉल्युबिलिटी. अगर वो ड्रग लिपिड सॉल्युबल होगी, only then it is going to cross the cell membrane, enter the cell and occupy its receptor. So the intracellular receptors are basically designed for the lipid soluble ligands. या तो ड्रग लिपिड सॉल्युबल हो, लाइक दी हॉर्मोन्स जो आपके स्टीरॉइड बेसिस के साथ होते हैं, फॉर एग्जांपल एल्डोस्टेरोन, कॉर्टिकोस्टेरॉइड्स, एंड्रोजेंस, तो या तो ऐसे हों जो लिपिड सॉल्युबल आपके फॉर्म बन जाती है. The second option is that the drug is so small in size that it can cross the cell membrane through the aqueous pores, which includes the thyroid hormone. उसके भी intracellular receptors होते हैं. So the examples of ligands for the intracellular receptors includes steroid hormones, vitamin D, thyroid hormone. अब हम अंडरस्टैंड कर सकते हैं इसको कि प्रोसेस क्या है. The process involves occupation of the intracellular receptor. चाहे वो न्यूक्लियस में हो, चाहे वो साइटोप्लाज्म में हो. The drug binds to the receptor and forms a hormone receptor or drug receptor complex. This complex will eventually move to the nucleus. अगर साइटोप्लाज्म के अंदर receptor है, the drug will bind to the receptor and this complex will move to nucleus. अगर receptor already nucleus में present है, the drug will move to the nucleus, bind to the receptor, and then that drug receptor complex will be present in the nucleus itself. When this drug receptor complex is formed, it is going to attach to a specific part of DNA, which is called hormone response element. Hormone receptor complex is the complex of the ligand and the receptor. Hormone response element is a part of the DNA to which this hormone receptor complex is going to bind. एक specific set of DNA को bind करेगा, उसकी transcription को activate करेगा. Now this transcription will lead to formation of messenger RNA. This messenger RNA will translocate to the cytoplasm. वहां जाके protein synthesis होगी. जिन proteins का उस DNA के specific set में transcription हुई होगी, वो proteins बनना शुरू हो जाएंगी. उन proteins को फिर modify किया जाएगा और जो भी आपका end product है वो बना दिया जाएगा. Now there are 2 to 3 points जिसको हम यहाँ से discuss कर सकते हैं. Firstly, It is not an immediate process. Hormone receptor complex बनना है, उसने hormone response element को activate करना है, transcription होनी है, messenger RNA बनना है, protein synthesis होना है. It cannot occur in seconds. ये immediately नहीं हो सकता. So these are generally slow acting receptors. At least 30 minutes to several hours are required for their action. Similarly, जब ये सारा एक process start हो गया, एक factory ने process start कर दी है, it cannot be stopped immediately. It is going to take time, again, at least 30 minutes to several hours to stop that process. So slow to start, slow to stop, not immediate. Number 2, it offers versatility. कितने diverse आपके पास फंक्शंस हैं, कौन सा स्पेसिफिक पार्ट ऑफ डीएनए एक्टिवेट हो रहा है, डिपेंडिंग कि किस टिश्यू में हो रहा है. किडनीज में एल्डोस्टेरोन किसी और पार्ट ऑफ डीएनए को एक्टिवेट करेगा, सोडियम चैनल्स बना देगा आगे से. कोर्टिसोल लिवर में किसी और पार्ट को एक्टिवेट करेगा, कोई एंजाइम बना देगा, ग्लूकोज फॉस्फोरिलेज और समथिंग जो मेटाबॉलिज्म में इन्वॉल्व होगी. दिस ऑफर्स सो मच डाइवर्सिटी कि आपके पास थोड़े से एक सेट मैकेनिज्म के साथ द डिफरेंट पार्ट्स ऑफ डीएनए कैन बी एक्टिवेटेड एंड मल्टीपल न्यूमरस फंक्शंस कैन बी परफॉर्म्ड. The next type of receptors are transmembrane enzymes. Again, as the name indicates, these are enzymes which are spanning the cell membrane. They are present through and through the cell membrane. जो part outside exposed है, that is an extracellular ligand binding domain. That is acting as a receptor. जो part intracellular है, that is acting as an enzyme. So it is one molecule, one protein molecule that is spanning the cell membrane and performing dual functions. The outside part is acting as a receptor while the inside part is acting as an enzyme. This enzyme is most commonly tyrosine kinase or serine kinase. Now a kinase is an enzyme that phosphorylates, and phosphorylation not always but generally leads to activation of the downstream molecules. Tyrosine kinase means that the enzyme is going to phosphorylate tyrosine residues. Serine kinase means that the enzyme is going to phosphorylate the serine residues in the amino acids or proteins which they encounter. The ligands for these receptors include insulin, epidermal growth factor, platelet-derived growth factor, and atrial natriuretic peptide. The mechanism is such that when a drug or ligand binds to the extracellular domain जो exposed है, it causes dimerization of two monomers. दो molecules जो अलग अलग पड़े होते हैं, addition or binding of a ligand to the extracellular domain causes dimerization of the receptor, which causes activation of the receptor. This dimerization promotes activation of the intracellular enzymatic domain. अब ये जब dimer बना, तो intracellular जो tyrosine kinase domain थी, वो activate हो गई within the same molecule. And this tyrosine kinase then causes phosphorylation of the downstream molecules, and that is how the signal is transmitted. These receptors are commonly downregulated अगर इनकी बहुत ज़्यादा excessive stimulation हो जाए. Cytokine receptors are very similar to the transmembrane enzymes. बहुत बेसिक सा छोटा सा डिफरेंस है. They are also transmembrane receptors. लेकिन हमारे पास जो ट्रांसमेम्ब्रेन एंजाइम्स थी वो एक मॉलिक्यूल के पास डबल फंक्शन के आउटसाइड रिसेप्टर था, इनसाइड एंजाइम थी. Cytokine receptors have an extracellular domain which is the receptor just like the transmembrane enzymes. But intracellularly, the molecule does not have an intrinsic enzymatic activity. Instead, एक separate enzyme है जो permanently attach कर दिया गया है इस cytokine receptor के साथ. That enzyme system is called JAK-STAT system, Janus kinases. Now these Janus kinases are slightly different from the tyrosine kinase activity. Transmembrane enzymes was one molecule with dual activity. बाह्य receptor and the transmembrane enzyme which is a tyrosine kinase. वो हर tyrosine residue को stimulate कर देती है phosphorylate करके. But here the cytokine receptors have an outside binding domain while the inside is permanently attached to Janus kinases which do not phosphorylate every substrate. They have a specific substrate that is called STAT molecule, signal transducer and activator of transcription. Now when a ligand binds to the cytokine molecule, again the monomers dimerize. They cause activation of the intracellular JAK-STAT system. Janus kinases are phosphorylated and in return they phosphorylate the signal transducers and activators of transcription STAT molecules. A STAT dimer is formed and as the name indicates, this dimer then moves to the nucleus because it is an activator of transcription. Transcription nucleus में होनी है. So the STAT molecule will move to nucleus and then will cause formation of the messenger RNAs through transcription and protein synthesis. So the downstream molecular mechanism of the cytokine receptors is also different from the transmembrane enzyme. Next are the ligand-gated ion channels. There are two kinds of ion channels. One are the voltage-gated ion channels, the other are the ligand-gated ion channels. Voltage-gated ion channels हमारे receptors की तरह act नहीं करते because they are controlled by voltage changes. Changes in the voltage or the resting membrane potential. Resting membrane potential अगर depolarization में आए तो कुछ channels open हो जाते हैं, अगर वो repolarize हो जाए, hyperpolarize हो जाए तो कुछ channels close हो जाते हैं. The opening and closure of the voltage gated channels is controlled through changes in the membrane potential. कोई drug नहीं आ सकती वहां पे unless कि कोई drug voltage potential को affect करे और वो channel को open या close करे. Directly नहीं act कर सकती. Directly कहाँ पे act करेंगी drugs? Ligand gated ion channels पे, where a ligand or a drug is going to bind to the extracellular site on the receptor, which is going to either cause opening or closure of that receptor, leading to either promoting the influx of certain ion or blocking the movement of certain ion. अब आप देखेंगे ये जो channels है ना, they're extremely quick. Milliseconds में ये act करते हैं. The drug is going to bind to the receptor is going to open like a gate for a few milliseconds, resulting in influx of a certain ion or efflux of a certain ion. And then the channel is going to close immediately. So, very fast acting, very quick acting and very short acting. These are commonly found in the central nervous system because हमारे जो reflexes हैं, motor function है, neurotransmission है, that is controlled through the very fast acting sodium channels mainly. So, हमारे पास examples क्या हैं ligand gated ion channels की? Nicotinic receptors to which acetylcholine binds and causes influx of sodium ions. Then we have the GABA receptors to which the GABA binds and causes influx of chloride ions. Then we have certain receptors to which the drug binds and causes opening of the potassium channels and efflux of potassium ions. So, depending upon different receptors, different ligands, different ion movement can be controlled through these receptors. Lastly, we have the G protein coupled receptors. These are also called serpentine receptors because of shape that is like a snake. 7 transmembrane loops form a snake-like structure, thereby called serpentine receptors. They are also called metabotropic receptors. Receptor G protein खुद नहीं है. It is a G protein coupled receptor. There is a receptor that is permanently attached to a G protein. Occupation of that receptor by a drug will cause activation of the G protein, and G protein will cause further downstream reactions. The sequence is such that whenever a drug binds to the receptor, it causes activation of the receptor. The receptor is coupled to the G protein. It causes activation of that G protein. Now we all know that the G protein has got 3 subunits. So there is an alpha subunit and there is a beta gamma subunit in a G protein. Whenever the receptor is occupied by the drug, it causes stimulation of the G protein. G protein के पास alpha subunit है और beta gamma subunit है. The alpha subunit gets phosphorylated, activated, and it detaches from the beta gamma subunit. अब beta gamma subunit के अपने function से alpha subunit which is activated causes further downstream activation of certain second messengers, which is a separate system. Now, what are those second messengers which are activated by the G protein? It depends upon the type of G protein. Majorly, we have got 3 types. वैसे तो there are about 13 to 14 types of G proteins that have been discovered so far. But at undergraduate level, we focus on 3 main types of G proteins, which are GS, GI, and GQ. GS is a stimulatory G protein. Activation of a receptor which is coupled to GS protein will cause activation of adenyl cyclase and formation of cyclic AMP. There is a GI protein which is inhibitory G protein. Activation of a receptor which is coupled to a GI protein will cause activation of the GI protein which is inhibitory in nature, and it will cause inhibition of adenyl cyclase and decrease in the production of cyclic AMP. GQ is a separate system. It causes activation of phospholipase C, which then forms inositol triphosphate, which is IP3, and diacylglycerol, which is DAG. Side by side, जब G protein, GS, GI, GQ are activating their subsets like adenyl cyclase or phospholipase C, जो beta gamma subunit it stays in the membrane and causes or modulates the effect of certain ion channels. GS protein causes opening of calcium channels. GI protein causes opening of potassium channels. These actions are mediated by the beta gamma subunit and not by the alpha subunit of that G protein. Moving into further detail, whenever GS is stimulated, it will cause stimulation of adenyl cyclase. Adenyl cyclase will result in formation of cyclic AMP from ATP. The cyclic AMP then activates protein kinase A. Protein kinase A causes phosphorylation of the downstream molecules. All of this is inhibited by GI protein. GI will cause inhibition of adenyl cyclase. Cyclic AMP will not be formed. Protein kinase A will not be formed, and the downstream mechanisms will not occur. On the other hand, the GQ protein stimulates phospholipase C. Phospholipase C breaks the membrane phospholipid phosphoinositol diphosphate. ये cell membrane में ही पड़ा होता है. It is broken down into IP3 and diacylglycerol. IP3 inositol triphosphate moves into the cytoplasm. It causes stimulation of calcium-mediated reactions. Diacylglycerol stays in the membrane and causes stimulation of protein kinase C. This protein kinase C then causes activation of the downstream molecules. This brings us to the point of second messengers. What are second messengers? Cyclic AMP क्या है? Cyclic GMP क्या है? Certain molecules which relay the message from the receptor to the effector. आपका जो drug है वो receptor के ऊपर message अपना transmit कर रही है. But जो effect है वो nucleus में से protein बन रही है. What is connecting this? The drug is not moving to the receptor to produce the effect unless it is an intracellular receptor. The in between जो आपके messengers हैं, they are called second messengers like cyclic AMP, cyclic GMP, calcium, IP3, DAG, all of these act as the second messengers. Cyclic AMP pathway is stimulated by the adenyl cyclase. Adenyl cyclase is stimulated by GS protein, inhibited by GI protein. Once stimulated, adenyl cyclase causes conversion of ATP into cyclic AMP. Cyclic AMP then causes activation of protein kinase A. Protein kinase A causes activation of certain catalytic molecules and causes phosphorylation of the downstream substances. IP3 and DAG pathway is mediated by phospholipase C. Phospholipase C converts PIP2 into IP3 and DAG. IP3 causes stimulation of calcium and calcium-mediated reactions, while DAG causes stimulation of protein kinase C, which then causes stimulation of certain phosphorylation and stimulation of certain downstream molecules. This is all about the signaling mechanisms and the second messengers that are involved in signal transduction. If you have any questions, please feel free to ask in the comments. Like, subscribe, and share. Thank you.